Ready? Okay, well, so welcome everybody. Today we'll talk about linear regression, something that we already started, but I'll go to start from the beginning there. This is supposed to be an applied course, so I actually, because this is the first real useful algorithm, I prepared a very simple application, and we go through that also. At a later point. So when I do practical stuff, I'll code in Python. Does everybody here know Python, or how many people know Python here? So pretty much everybody, almost everybody. Okay, so that's good. Whatever, I mean, the syntax is really easy. The scripts are usually very short, and yeah, I mean, so I'll just show you whatever is not known. Okay, so a quick summary of week. So today I'll repeat what we've done last time, which is the Bayesian part of the Gaussian, very briefly. Summarize what we've done last week, and then go to linear regression. So Bayesian inference builds on the sum rule and the product rule. To get Bayes' theorem, we said that that given the joint distribution and the marginal distribution, where the joint distribution is written as the conditional of one given the other times the marginal of the other, using the chain rule, together with the product rule, we can get Bayes' theorem. And in Bayesian machine learning, we'll use Bayes' theorem in the following way. So we have a model where, well, in maximum likelihood estimation, we used the likelihood, maximized the likelihood with respect to the parameters, and those were our estimates. But in Bayesian inference, we treat the parameters as random variables instead of getting a point estimate for them, and put a prior distribution on them. The prior distribution summarizes our prior belief on the parameters, how they could look like over all different data sets that we might be interested in our domain. And well, the posterior, which is the probability of the parameter now given the data, is proportional to the probability of the data given the parameters, so the likelihood of the parameters times the prior of the parameters. And this, in order to get a conditional distribution, we need to know. So this is not the joint distribution of the data and the parameters. In order to get the conditional distribution of the parameters, we need to normalize it by the probability of the data, which is, again, using the sum rule, the sum or the integral over the joint distribution over these two. And then again, using the chain rule, we can just plug this in for every possible parameter and then integrate out that parameter. So in the Bayesian, in the Gaussian model, the likelihood is a normal distribution. And well, for example, with a normal prior on the mean, and well, then the posterior is just proportional to the product of the two. And this kind of learned about map estimation, which is kind of between frequentist estimation and the Bayesian part, where we still have a prior, but we still just maximize the posterior. In order to maximize the posterior, we only need to look at that part here on top of the ratio, because probability of the data doesn't depend on the parameters. So if we maximize that, it doesn't matter. Well, we learned that for when estimating the parameters of the mean, we usually work with the log of the posterior because it's easier. Doesn't necessarily have to be, but it's often the case. Take the derivative, maximize, and then we get the map estimates, which is kind of a weighted average of well, the prior mean of our mean and an average over the data. So, and we can basically think of the ratio of the variances of the prior and of the data as kind of a regularization parameter, because this kind of tunes how much we rely on our observations and how much on the prior.
Can you talk a little bit more about the regularization parameter? What is that actually? How does that help?
How does it help? Well, so I mean, in the limit of infinite data, it doesn't matter, huh? No matter what it is. And for finite amounts of data, basically, it tells us, well, whether we want to go far away from our prior or not. So basically, and well, it's not clear how to set that yet, but as I said, it's kind of, I mean, the prior so far kind of fell from the sky, but we will talk about methods and how to set that from data. For example, something that is yet in between, still in between Bayesian inference and estimation is, well, if we have our marginal distribution over the data, which is the probability of the data given the prior times the prior, the integral over all the possible parameters, then this marginal distribution still depends on the parameters of the prior, right? It still depends on, for example, on this mean zero. And we can again view this, for example, as a likelihood, and then do maximum likelihood estimation on that, which is a bit further away from the data than standard maximum likelihood estimation, but it's still a form of maximum likelihood estimation. Well, it's hopefully not as so the problem with maximum likelihood is usually that for small amounts of data, the parameters vary a lot. So I give you a different data set of a rather small sample size, and you will get a completely different parameter. That's kind of the issue. And if I set this increase this regularization, then my answers will vary less and less for finite amounts of data.
So it's something that you set like beforehand?
Yes. Yeah. I mean,
just depending on like whatever.
Well, I mean, as I said, you could do this kind of hack, which is done often in practice, and that is maximize the marginal likelihood with respect to it, which in that case you actually didn't specify any prior parameters. It's kind of you still use only the data, or well, if you have, for example, a different experiment beforehand, we talked about this way that kind of our posterior summarizes everything that we need about it. Summarizes both our prior and the likelihood. We can use the posterior from a different experiment again as a prior for a new experiment. Or there are methods like cross-validation, for example, where we'll talk about that later, but one very shorthand is kind of, it uses prediction. I mean, you can use these models also to predict the value or the likelihood. Basically, for a given data set, you can compute the probability of the data under this data set. That's the likelihood. And if you take the marginal likelihood, for example, apply it to learn it on one data set and apply it to another data set, then you can, for example, choose the parameter that if you maximize on the one parameter, on the one data set, you get a high likelihood on the other one. So basically the procedure is have two data sets, maximize on the one and predict the other, or no, not maximize on the one. Choose the prior on the one data set. Then compute the posterior or compute the maximum posteriori estimate or something like that. Use that on the new data sets and take the argmax of those with respect to the prior parameters. So these are all kind of methods that go away from the, I mean, if you talk to real Bayesians who believe in this like very deeply, then for them the most clean thing is to be absolutely subjective. You have a prior which, if you haven't seen anything, is probably pretty uninformative in the sense that it lets the parameter free move very often, but if they already have seen some data sets, then it becomes more informative, more peaked, and you would only use that. But in reality, we kind of have to take bits and pieces, combine them in a way that works well. Okay, so well, in order to do Bayesian inference, we said we use data again, use a model, can use a model as a hypothesis, and well, this gives us parameters, and then we, so the likelihood is now the product of all the data points under this model, and we combine it with a prior distribution. Get something that is proportional to a posterior. And if we want to do Bayesian inference using, well, it just happens that if we use a Gaussian likelihood with a Gaussian prior, that we can compute the posterior pretty easily, and we actually don't have to take into account all the normalization constants, because the posterior, again, is a Gaussian, and we can just it's a Gaussian in the parameter, which in this case is the mean. So we can just find a Gaussian by singling out the mean from that form, right? And then find the parameters that correspond to a Gaussian. Yeah.
So you sent out the email this morning saying that problem 1b was intractable. So does that basically mean that it's computationally difficult?
Well, I mean,
like, why do we care about being able to combine it into one distribution instead of just having two distributions multiplied by each other or whatever?
Okay, so for example, if we want, so that's one nice thing about, for example, maximum or posterior estimation. We don't need to compute the posterior. There, if we only want to maximize, then you can only always work with something that is proportional. But if we want the posterior distribution, then we want something that sums up to 1. So we want a distribution, actually. Well, we can always use, being able to write out that distribution in closed form is a nice property because it saves us a lot of headaches. And there are other methods to do that, for example, Markov chain Monte Carlo methods, basically, which corresponds to randomly draw samples from the prior. Basically, let a machine just draw parameters from the prior, plug them in, and evaluate the whole thing. Or evaluate something that is proportional to the posterior, namely the joint distribution over the two. And then, in the end, well, if you had 100,000 samples of that, you can basically approximate you kind of get the shape of something that is proportional to the posterior, and well, use that kind of, if you sum all of these together or average over all of these posterior values, then you can normalize your distribution empirically.
So basically, intractable means it's difficult to calculate all the constants.
Right.
Oh.
Yeah. You cannot just write it down. That's what I mean with intractable. Intractable does not mean you cannot do it. Just means you cannot write it down. Yeah, I mean in one dimension, so far everything was in 1 or 2 dimensions. I mean that is a 1 or 2 dimensional problem. Pretty much everything is easy. But if you go to higher dimensional problems, for example, if you have a neural network where you have multiple layers and each, well, you have all the neurons and each neuron has a set of parameters computing the posterior over those. Forget it. Right? Or even doing an exhaustive Markov chain Monte Carlo with exhaustive I mean, you cannot do anything nearly exhaustive even for those. Hello. Okay, so the nice thing sometimes if you use these conjugate priors, that we can get the posterior enclosed form. Yeah, so, well, here we looked at the posterior parameters of that, where we saw that, well, if we don't have any data, then we basically have the prior. If we have an infinite amount of data, then we are at the maximum likelihood estimate. And, well, we also saw that if we increase the sample size, then this becomes more and more peaked. And
would you mind explaining that graph a little bit more? I just didn't quite get why is it peaking over in the right more? What is it?
Well, it's just there is. I mean, the data comes from some true distribution, which we don't know,
Right.
which has a mean. And well, now we compute the kind of try to estimate that mean. In the beginning, we don't have any information about it, so we only have our prior.
Oh, so this is just
now
an
we
example. It's not saying like this is always where it will peak.
No, it's not where it always peaks. It's just one example. That the mean is probably somewhere around 1, or well, must be somewhere where the posterior actually has some mass.
Okay, that makes sense.
And yeah, just says that the posterior converges to the mean of the data, and becomes more and more peaked, so more and more certain about where this is. Well, we talked about conjugate priors, where, well, for a conjugate prior is a prior for a particular likelihood if the prior has the same functional form as the posterior. So if we know the prior, we can write down the prior, we can write down the posterior. The only thing that's different is the parameters. And, well, these conjugate priors exist for a group of distributions, where there's the exponential family, which can always be written down as this. And, well, if you look at, for example, the Gaussian, then we can, well, in the traditional form, it doesn't look like this, but we can do the math and wiggle around with it, and then we find a new form of the Gaussian where the parameters in the exponential family form are the ratio of the mean and the variance, and something that is minus 1 over 2 times the variance, for example. I guess there are also different ways to do the same thing. And, well, some functions of the data where they don't necessarily have to depend on the data, but they may. Well, plug that in, we get something that looks at that form for the Gaussian. So that shows that the Gaussian is an exponential family, or is a member of the exponential family. Well, so the exponential family kind of gives us, as long as we always choose distributions that belong to the exponential family, we can also find a conjugate prior for it. That's what it means. Doesn't necessarily mean that the conjugate prior is a good prior. It just means that we can choose a prior that is tractable.
What
Construct is basically choose the conjugate prior, and
You pick the distribution that you want.
well, I mean, if I choose a prior, I can choose the distribution that I want but I have to take a specific functional form, which is the conjugate prior. So that I cannot choose. If I want the conjugate prior, then I'm limited to a specific functional form.
Are there any cases where a conjugate prior is not a good choice? I mean, famously.
Well, usually the conjugate prior makes sense. I mean, there's a reason why it's the conjugate prior. It very often makes sense. Sure. It's a good question. I think about it some more. Well, I mean, for example, I mean, you have seen the normal gamma prior, right? That's obviously was in the email today. Where, well, let's go to that. I mean, it's now actually on that form, on that. Well, where basically now the mean depends. Well, the mean and the variance now. So the variance of the mean now depends on the variance of the data. Doesn't necessarily have to be a good thing. I mean, it's really if you specify these priors.
This is what you changed, right? This is changed from last time.
This is changed, yeah. So the joint prior over the two is now probability of the mean given the. So the probability of the mean is a function of the variance in this case.
Yeah.
So it's this here.
Because in the normal, yeah, you had that statement like about how they're independent. And
Yes.
that's no longer true anymore.
Not no longer true anymore. It's not the theory changed, it's just that I had to type. Okay. Okay, and we also said, okay, so if we use this conjugate priors, then we can use this idea of sequential learning. We could also do it with non-conjugate priors, but it's harder to do. If you use conjugate priors, then we can just update the parameters and we can just read it off given the data. So we have a number of independent data sets. Now we have a prior in the beginning over the parameters and the likelihood, and now the posterior, seeing one data set, we can just compute like that. And then we can use that posterior as a prior in for the next data set, and so on and so forth. Yeah?
So, talking about priors, I don't know if this is maybe a discussion better off for another time, but if you're just given some data and you have no idea what the parameters are or how they're possibly distributed, how do you choose a prior? How do you choose a distribution for the prior?
Well, there is a lot of work on how to choose very uninformative priors. Priors that don't influence your data as much as possible. I mean, okay, never say there's say uninformative prior, and prior is always to some extent informative, and it's supposed to be. It's supposed to kind of regularize your finite data estimation or inference. But a uniform distribution, for example, is pretty. I mean, you can, for example, with the Gaussian, you can construct something that looks like a uniform distribution by making the variance infinite. Then you've got something that is completely flat. And if you look at that, then actually the posterior distribution or the posterior mean will look very very similar to the maximum likelihood estimate. Often there are like small changes, but.
So do you just try to choose something that tries to like allow your parameter to vary more, or to like have, well, you have some initial estimate of your parameter, right? So then you're trying to pick a distribution that makes that initial guess influence your ultimate outcome less? Like Or do we just
I-
choose a prior that lends itself to like tractability?
So what I do in practice is usually I choose a prior either that is very uninformative in the sense that it allows a large range of values, or I choose a prior such that well, the marginal likelihood is good, is well predictive of the data, so that it represents the data well. So I usually use these kind of empirical ways to choose a prior.
So is this related to that graph that I just asked you about a little bit ago? Like, would this be something like the first prior is just like normal 0, 1, but then the second time around,
Yes.
like in the second sequence, you're saying, all right, now we know it's a little bit more linear. And
Yes, I mean it's
more
just,
data.
I mean it's just a way to compute this. I mean, of course, you can just merge all of these data sets together and just do this step, and then you're already here, right? I mean, but you can also just, as a data point comes in, update your posterior, and then you would also could.
So if you applied like this method to 10 data points, you get something like that graph that you showed?
Yes. Yeah. If you plot each of those posteriors, then you get exactly that. Yeah. Uh-huh.
So in the real data, so you have some observations, and what type of method could you use to actually estimate the distribution of your observed data? So this will be your prior, right? So I'm just wondering what type of method you are using in practice to estimate the distribution. And maybe it's like a normal distribution or like a gamma distribution, and do you have like a p-value to indicate that, or you just have confidence that?
So, well, if you're Bayesian, then or well, if you're Bayesian in general, the marginal likelihood gives you a very good way what the marginal likelihood tells you is how good does my likelihood and my prior distribution. So my model is basically consists of a likelihood and a prior. How well does this reflect my data? So if I compute, if I do the integral, yeah, the number that comes out of it is basically the probability of the data under my model. That's the marginal likelihood. Choosing a model that gets a high value for that is good. So if you have a very complex model, then for the best parameter, you might get a very high likelihood. So if you have a model that let's say we have a Gaussian model for n data points, but we now specify a single variance for each data point, right? So each data point may have its own, or it may have its own mean. Let's say it like that. Then, well, we can, if we maximize that likelihood, we can pretty well represent our data, because we can just set the mean of each of those data points to the actual observation. But once we integrate out all these means, we have to integrate over a whole lot of values, because there are many data sets that could be produced by such a model. And for that reason, the marginal likelihood won't be very high, because it has to kind of spread out all its mass, right? I mean, it's something that it's a very complex model. It can represent many data sets, and for that reason, it kind of has to spread out its mass over all the possible data sets. If you can represent more data sets, so if you have a more complex model that can represent more data sets, then for each specific data set, your marginal likelihood will be not very high. Is that, for for example, yeah, I mean, if you only well, we will talk about that on another day. I have some material on that. But the marginal likelihood tells you what's the probability of my model. And typically, if you have a more complex model, there's kind of this trade-off, complexity of the model. And if you have a very simple model, then probably the likelihood for each set of parameters will not be very high. The marginal likelihood might still be higher than a more complex model, where the likelihood where the maximum likelihood solution is very high, but you have also many parameters that give a very low likelihood of the data. Okay, but that was too much of a detour now. Okay, so we can get this sort of sequential estimation. So that was week 1. Where we talked about probability theory and uncertainty, sum rule, product rule, Bayes theorem. Talked about Bayesian learning, entropy, maximum likelihood estimation, and Bayesian inference. I had the example of the Gaussian. And now we come to the new stuff. Start out with linear regression. We already touched it a little bit last week, and we'll talk about. I'll talk about an example, which is genome association studies, and well, there was the question of can we get significance estimates for a model, like a p-value or so. In order to get there, we'll talk about some more properties of normal distributions in order to compute something like that, and talk more about these regularization methods. All right, I have to thank Ollie. So about 1/6 of the material is from a course that we have given before. So that's probably the slides without typos. The other 5 out of 6 are the ones that we are proofreading that now. Okay, so regression. So in regression, the idea is that given a data set, we want to find a function, function f, which we call a regressor that depends on parameters and on the data. And now, well, the data comes in pairs where we have inputs x, outputs y. And well, we plug in the inputs and the parameters into a function that spits out a value, and we model our data, our y's, our outputs, as this function plus noise. And the noise, for example, is Gaussian, typically. It's just the way it's typically done. It doesn't have to be Gaussian. Then, well, the likelihood is now a function of x. So x is always given, the inputs. Those are assumed observed. It's now a distribution over the outputs. So the likelihood is always computed over the outputs, not over the inputs. And the mean of the distribution is the function, and well, we have a variance of the error, where the variance is just the variance of the noise. And if we choose the function to be linear, then well, we just have a linear function, so the parameters are now just multiplied to our inputs, where the inputs are in the form of vectors. Well, we also have kind of an offset, which is a mean, but we can always drop it from our equations, just doing this little trick of appending a 1 to each input. vector. If you have a 1 at the end of each input vector, then we just also make our betas 1 entry longer and put in the C, this constant, into the beta.
Where does sigma come? Where does the variance come from? Like, even though going back to
Okay, so the variance comes from the function, again, I mean, because we have a Gaussian likelihood, the function doesn't explain everything in our outputs. There's this kind of, well, it's a function plus something like measurement noise, independent noise, and well, the sigma is just the variance of that measurement noise. Okay, a way to represent this with a graphical model, we basically have this plate here, where we said the plate just takes everything that is in the plate and replicates it that many times. So we have n observations of x and y, and we model y as a function of x and the parameters, where the parameters are outside the plate because they are the same for each of those data samples, and well, y also has a variance, which is the noise variance. And well, again, nodes are these dots, and then we have dependencies between the nodes in the form of arcs and arrows. And directed arrow means that there's kind of a dependence that goes in this direction.
Can you quickly explain this column of ones?
The column of ones, okay. So say
Actually, maybe for the video it would be better to.
okay.
Yeah, this is great.
It's useful. Okay. Say I have x1, x2. So that is and the y, right? Sorry. That y equals x1 beta 1 plus x2 beta 2 plus c plus epsilon. Then, well, that's basically, this is a vector now. I represent it as a vector, where the vector is, well, this is just like a scalar, so it's 1 times 2, x1, x2, and beta is also a vector, 2 times 1. And now if I also want the c in there, I just put a 1 in here, and I put c into my beta vector. Right?
So you assume that the noise is normally distributed, right?
Yes, this is a model assumption.
Yeah, yeah. Does that mean that y is also normally distributed? What's the correlation between?
Yes, y is normally distributed given x and beta. So given the regression function, y is normally distributed.
But otherwise
Well, it basically means that y minus the function, which is the epsilon, is normally distributed.
But y itself could not be normal.
Yes, I mean if I say, well, there's a very, I can give a very simple example. Say x can take on 2 values, 0 and 1. And well, so 0, 1, and now I've got some function, some function. Well, now my y is here. So if you, so this is the function. These are my x values. Then I have basically, if I plot y minus the function, I get a single Gaussian, because well, this is a Gaussian with the same variance as this, right? But if I plot both together, so if I just take a histogram over the y's, then the histogram will look like this. So it's very non-Gaussian. While if I subtract the function, it will look like this. Right?
What are you subtracting? You're subtracting that line?
Well, okay, so that is x, that is y, right? This is now f of x and the parameters. So basically, in this case, it's x times the weight plus plus the offset. I'll say there's no offset for simplicity. So this is now 0. Well, no, there's an offset because I haven't painted a 0 here. Okay, so it's a nice thing about a board. You can just change your coordinate system. That it's fine. All right. So now this is 0. This is 1. So basically, it means that my beta in this case is just 1, right? So given that that the input, the x of a sample is 1, the mean of your sample is 1. Given that the input of the sample x is 0, then the mean is 0. That's what this means. But it does not mean that y is around 0. Around 0 is Gaussian distributed. If I don't use this function, as I said, I get two bumps where one is at 0, one is at 1. If I subtract the function, then everything will be around 0. Right? Is that clear?
So why do we assume that the distribution or that the standard deviation, for example, for the two populations are the same? After the 0 and 1, like why do we assume that they just happen to have the same variance there?
Again, this is a modeling assumption. So it doesn't have to be true. If it's not true, then it's probably not a good function, and we should change our model. So basically the model that you write down is always, you make a lot of assumptions. And if the assumptions are bad, then your model inference might be bad. Usually it's good to write down a simple model, because, well, if you have a simple model, then you have very few parameters, and hopefully, you're not overly confident running into a wrong direction. But it's just a model assumption. You could also model the variance as a function of the mean, right? That's basically what you would mean is that if your regression function says, well, if your regressor is large, then you also get a larger variance. I mean, that's something that makes sense, right? In some cases. Doesn't necessarily mean that it makes sense. For example, if you want to model height as a function of age, so, right? Say between, we don't consider that people also get shorter, but say they only grow. But it does make sense because the function at a given age depends on the height at the previous age, so kind of noise adds up over time, right? So if the noise gets larger over time. In that case, so there probably such a model would not be ideal. Yeah. Okay. Well, how do we find the parameters? First, well, we take the likelihood, and we again take the log likelihood. Now, if we look at this log likelihood, we have again the Gaussian part where the mean is now this linear function. And if you look at the linear function, we again have a sum of squares estimation. So if we want to maximize this likelihood, we have to minimize, because there's a minus this sum of squares term here. And well, this means that least squares estimation. So least squares regression is the same as maximum likelihood estimation in this model. So whenever we do a least squares regression, as something that many of you have heard before, I'm very sure, and you're actually writing down this Gaussian likelihood and maximizing it implicitly. And this is the same thing. So we have our data points, and the regression line, if we do maximum likelihood estimation that we fit, is the one that to all of these data points, to all the y's, has the smallest squared deviation. Now, how do the parameters look? In order to maximize this, we only need to look at the part that actually contains the parameters, which is the squared term. Actually, this should be a beta. The notation is a little bit messed up. But we take the derivative with a single beta, and well, if it's multiple, we want to jointly maximize it. So we use a generalization of the derivative, which is the gradient. So if you take the derivative with respect to each of those parameters, so beta 1, beta 2, and well, beta 0, or c, and just concatenate them into a vector, then you get something that is a gradient, and the vector has the same dimensions as the vector beta. Now, how does the gradient look? Well, we take the derivative of this, well, one term in the sum, so we get 2 times the part in the sum, times the derivative of that, which is then the beta I. We plug all of these into a vector, and we get well, the inner part times the whole derivative of that part, which is x. So how does the maximum look? Can you maximize this function? Well, let's just do the math. We have to basically get the beta on one side. So let's take the x, multiply it in. So we have x transpose times yn in the sum for each term, and xn transpose, and xn transpose times beta. Then we put that part on one side. That doesn't depend on beta, and then we have here an xx, so a square of x. We take the inverse of that, multiply both sides by the inverse to get rid of it, and then we get this part here, so which is take all the x's, put them into a vector, and we get this part which is the inverse times x transpose times y, where this part is the so-called pseudo-inverse of x. And well, we define the matrix X as just putting all the vectors axes that we have, so all the inputs into a single matrix. And this is just the maximum likelihood parameter. Now, we already know how to do a least squares regression. So it's really simple to write that down. And let's apply it to, well, actually, because we get to the application, let's do a short pause here. We kind of X as U, or U is a matrix of eigenvectors of X transpose, and the matrix times S, where S is a diagonal matrix, and the matrix V transpose, where V holds the eigenvectors of X transpose X. And S is the singular values. If you invert all the non-zero singular values, you also get the Moore-Penrose pseudo inverse. I mean, it's many ways to compute the same thing. So the maximum likelihood estimator is just a pseudo inverse times the vector of y's. Yeah. Okay. So any more questions to pseudo inverses? Yeah.
So I'm trying to get like a sort of feel for what the pseudo inverse really is. I mean, we know the properties, but what does it really give us, like in terms of transformations and stuff?
I can't really answer this question right now. What does it give us? Well, I mean, it's an inverse for matrices that are not invertible. It's kind of a way to make up an inverse. It looks like something, of course, in order to have something that generalizes an inverse, for the case of an invertible matrix, it has to reduce to an inverse. So the Moore-Penrose pseudo inverse of an invertible matrix is the inverse. And, well, all these properties are also, of course, fulfilled by inverses, which is basically, if A is the pseudo inverse of X, then AXA is A, but also X is the Moore-Penrose pseudo inverse of A. That's also what it means. So basically, if you take X, AX, you get X. And I tried to think what the other properties were. I don't have them at hand. I mean, if you want, I can add a slide on pseudo inverses. But I mean, we'll meet them later again, maybe at a time where they fit in. Let's just take it now as a given that we can compute this thing. Okay, so let's come to an application of linear regression, namely genome metacization studies. So we said we have the genetics for the individuals, and so we measure all the mutations in the genome, or a set of mutations for each of those along the genome. So it's positions in the genome where one individual is different than another individual, and we do that for all the individuals. We see whether they have the kind of the wild type, which is the kind of this, say, for example, here, if most of the individuals here have, most of the individuals have blue, which they don't have because it's 6 versus 6, then we call that the wild type, and we would call red or the reference, and red, for example, would be a mutation, right? So we kind of see which individuals have at a position the reference allele and which have the mutation. And these usually come in the form of single nucleotide polymorphisms. So it's single, I mean, you have the the DNA, which is ACTG. And so it's nucleotides, so it's single nucleotides that are polymorphic between the individuals, so that vary between the individuals. It's single positions in the genome that vary. And the phenotype, for example, the height of these plants, because we're in the linear regressions case, we want quantitative phenotypes typically, so something that we can represent by a number. And the goal is to find some SNPs, markers. We call them markers because they kind of stand as a marker for the region that they're in the genome. So they're somewhere located on the chromosome, and well, they mark it. Because if you measure two neighboring markers, then there's a strong correlation between those. And so if we, for example, have this plot where we plot the position on the chromosome and some measure of statistical association between the phenotype and so each of those dots is basically a measure of association of a single nucleotide polymorphism of a single SNP and the phenotype. And we see that, well, here there's some peak, so there's probably something that is associated, but also around this peak there's a lot going on. So these peaks are not just one SNP, they are broader, and that's why we can use them as markers, because they are all correlated in kind of to their neighbors, and for that reason, even if we don't have the true causal mutation in the genome, I mean the causal mutation might, for example, be a deletion that, for example, some part of a gene is missing or so, and which we didn't measure in the beginning, but if you have such a single nucleotide polymorphism marker in the close region, closely located to that gene, it will kind of, because it's correlated, tell us something about the location of that causal mutation. And we'll use linear regression to do this sort of analysis. Well, the genotype denotes the genetic state of an individual, and that is our input X. And well, for individual N, we call it the XN. No? The phenotype is the state of the trait. So it's, for example, something is red or blue, in this case, red or white. And we call it Y, which is the output. So try to fit a regression function from X to Y. A locus, I just want to introduce all these words, not that I throw around with them and you don't know what they are. Locus is kind of a position on the chromosome, and it's a limited region. And well, we use these SNPs as markers for loci. So they represent loci. The SNP is not the loci, but basically, if I call a SNP a locus, then it's kind of the place where the SNP sits. And an allele is the genetic state of a locus. So for a SNP, this would be its reference or wild type, or it's the mutation, the minor allele. Minor allele because we observe it. Major allele because it's the reference, because we observe it more often. Minor allele is the mutation because we don't observe it very often in the data set. So typically we have an organism has some sort of chromosomes, and if we say that an organism is haploid, then this means that it only has a single set of chromosomes. So humans, for example, they have two sets of chromosomes, but there are some other organisms that only have a single set. Or we also do some slight abuse of mutation. We also call an organism often haploid if all the chromosomes are identical. That is the case for example, Arabidopsis thaliana. Our sperm cells are also examples because they only have a single chromosome set. or if you have, for example, some lab mice, usually what we do with lab mice is that because we want to single out a certain type of genetics, we basically, well, all their parents are clones of their kids, and so they're all clones. They're all completely the same. They're usually so also haploid in that sense, because both the chromosomes they have 2 chromosomes, but they're usually identical due to inbreeding. We call it diploid if there are 2 independent chromosomes that are inherited independently from the parents. And well, there's not just haploid or diploid. I mean, we won't touch it, but there are crazy things. For example, a sugarcane plant can have many different sets of chromosomes, and it can even vary between cells or between individuals. I'm not sure. I think it's between individuals actually. So one individual, even they're the same species, but one individual has 10 chromosomes, the other one has 6 or so. Right? So it's quite funky. Well, the haplotype well, denotes the individual state of a single set of chromosomes. So there's a parental for a single chromosome. It's basically there's a parental or a maternal haplotype type, and we call the locus heterozygous if the 2 chromosomes differ at a locus. Right. So if so you can have for a SNP, if that SNP is well, if it's in a diploid organism, it can either be twice the reference, one time the reference, one time the mutation, or you can have twice the mutation. So we call it heterozygous if you have one time the mutation, one time the reference. We'll denote, well, now we have to make up numbers from SNPs to numbers in order to use it in a regression. We call that a 1. And if you have something that is homozygous, that means that the two copies of the SNP match. So it can either be twice the reference or twice the mutation. And well, major allele the reference, minor allele the mutation. If you have twice the major allele, then we call it a 0. Twice the minor allele, we call it a 2. And well, in summary, basically, we count the number of mutations at a position, right? Now, we use a regression function where the phenotype is a linear function of the SNP, of a single SNP and a mean. So it's typically we have the model where we have the X as a single SNP. So SNP times beta plus beta 0 plus noise, right? And then we call basically make out of this an X, which is the SNP, and a 1, and beta has now 2 entries, beta 1 and beta 0. Now we'll look at a specific study. We use Arabidopsis thaliana for that. So let me just quickly tell you what Arabidopsis thaliana is. It's basically this little plant here. Arabidopsis grows, it's very robust. It's basically, as you can see here, it comes out of pavement, for example, here. So many places you probably see it, and it just grows out of some places. it's distributed over the whole globe. So everywhere where there's color, we can find some Arabidopsis. It's naturally from here, and the blue areas are where it's gone to. It travels far because the seeds they just hop on the plane, and then from here to here, and then they spread out pretty quickly, and you have them everywhere. And well, so basically, these are kind of ancestors of these. It's been studied for a long time, so you can think of it kind of as a lab rat among the plants. And one interesting thing is it's mostly self-fertilizing. So the plant has both male and female organs, and usually, for example, if it's on the pavement like this, if it wants to reproduce, it has to self-fertilize, right? So pretty much all of it is homozygous. And actually for the data set that we have, everything is homozygous. So homozygous makes it easier. We don't need a 0, 1, or 2. We can just work with 0 and 1. Right? And Arabidopsis is a model for flowering in plants in general. So that is really well studied. We know we can measure pretty much where all the involved proteins and go, and in what parts of the flower the genes are expressed, and so on and so forth, and can measure that over time. Well, we just use 0 and 1. We'll use 0 and 1. Okay, the data that we use is from a Nature Genetics paper, et al. The phenotype is flowering time at 10 degrees Celsius in days. So we just take some plant, we put it into a pot, and now we wait until it flowers. So it grows, at some point it will flower, and we measure the number of days. The genotype, well, is our matrix X, 214,000 SNPs. And all of them are homozygous, so that's why we can work with 0 and 1 in this case. We have 194 of these plants, and now we'll do a GWAS with them. Okay, so this. I did this in an IPython notebook, but I didn't know how to run an IPython notebook on these machines here, so I copied it over to a PowerPoint slide. So it's just some imports in the beginning. It's pretty boring. We have some data file. Well, this is the fifth phenotype. It's the flowering time at 10 degrees Celsius. That's the name of the thing. We have some matrix X, which we load. So it's the genotypes. You get the positions. The positions actually has 3 entries here. It's the chromosome. So in this case, it's 5 chromosomes. So it's a number between 1 and 5 for each of those SNPs. We have the position on the chromosome. So that's just an integer. And then we have the cumulative positions. The positions for each chromosome, you basically start counting over again. So if you want to put everything on one plot, for example, if you want positions that don't overlap, we have to kind of use basically if one chromosome it goes up to position 1 million, then for the next chromosome we cannot start over at 1 again, so we have to start with 1,000,001. That's the third thing here. This is the model, right? So y equals x beta plus epsilon, where we use the maximum likelihood estimation. And that code is here just to show you that the implementation is really, really simple. I don't know, is this large enough to read? Okay, so, if we want an offset, basically we do exactly what this trick that I just said. We just append, we take our x's, and we create a vector of ones of the length n, where n is the number of individuals, and we just append it to x. Right? So we have our matrix X, This, I mean, the kind of, everybody used to this kind of mathy notation here. Now we have to compute this part. So this is now x including, so before it was just the snip. Now it is the snip plus the offset part. Now we compute our, this part here. For that we compute x transpose x. So it's just x transpose dot x, so it's a dot product. That's NumPy, it's really elegant and nice. Take the inverse of that. We do the same thing between x and y. So we don't compute the pseudo-inverse and then that, but in this case we compute the dot product of that and the inverse of that, and then take the product of those. So, and then we've already coded the linear regression. We can also then measure the error that by just taking y minus x times beta. And then we return that. So it's just a couple of lines. Well, we run it, we take our x's, we run it for each of the snips. Get our beta, and now we take our big X, which is 240,000, but we only use a single snip at a time, so that's why we have a for loop over all the snips, and we just input that as the X in the linear regression function that we had before, and keep all the betas and the residuals. Well, okay, so if we want to plot this, and the betas, they can get positive or negative. We take the squares. And now what we plot is well, just for chromosome 1, we plot the position on chromosome 1 versus the square root of the betas. And then we get a plot that almost looks like the one that I've showed you before, right? Yeah?
So why don't you just use the absolute value of beta?
Well, if I use the absolute value, then you wouldn't see much. I mean, if I square it, I see a larger difference. So, but we could have used the absolute value. The plot would have been very boring, and yeah. But typically, we'll see that also in the end. Once we go to the statistics, we'll do the statistics usually with something that is related to the squared. Well, let's do something else. I mean, one observation is that, well, if one of the SNPs, say one of the SNPs has kind of, if you take all the individuals, it just has a larger variance than another SNP. Then what happens is that basically, I mean, you want to explain all the variance that you have in Y, but if now you can basically, if you just take the same SNP, you increase the variance in the SNP, just multiply it by a constant, for example, then you can account for that. So if you compute your maximum likelihood beta, the beta will become smaller. Right? Now, for that reason, I plot something else, which is well, the betas times the standard deviation of x to account for that. And then take the square of that again. It looks very similar to the first plot, just that the peaks maybe are a little bit more sharp. So, and you again observe that, well, if there's a peak, there's not just one thing coming up, but many things in a region. Well, we can also pull out the maximum of those squared betas, so the one that has the maximum squared values. And now we plot the regression function for that. So the regression function for that is this red line. And we separate out the individuals actually by that SNP. So we take all the individuals and we plot the SNP of those individuals versus their phenotype, and then we see that, well, there's some separation here. And the regression line right fits through these. So that's all that the linear regression does here. Well, it looks like there is a difference between individuals that have the reference and some that have the mutation. It's also on the right side. There are fewer because that's the mutation and that's the reference. basically. The question is now, is this real? Can we now write a paper that we found a SNP that where some individuals take longer to flower on average than if we don't have that mutation? That's kind of, we'll need statistics for that. This is just code. What it does is just it plots kind of we give it some values and it plots a histogram of those values and fits a normal distribution to that. The fitting of the normal distribution is just the maximum likelihood fit of the normal distribution that we've talked about before. So we do that with our betas. Just we take all the regression weights, all those the betas for the SNP, those beta 1s. Actually, here it's beta 0s. And well, we plot the histogram. We see that it looks a little bit Gaussian distributed, but a little bit slanted also, right? And we do the same thing for our beta times the standard deviation of x, and the same thing is true. So it's kind of Gaussian distributed, but not completely. The other thing that I've noted in these Gaussian plots here are 2 times the standard deviation. So the area between, for the Gaussian, the area between the mean minus 2 times the standard deviation here and the mean plus 2 times the standard deviation is 95% of the mass. So here we have 2.5% of the mass. Here we have another 2.5% of the mass. Well, let's just do, so we want to see what happens. Is there something real? Is there some information? So for comparison, we'll now do something funny. We'll just create a permutation of length n, and then we just kind of create a random phenotype. So instead of using the real phenotype, we use the phenotype values and we just randomly permute this. So before we had kind of joint observations of your SNP and the phenotype for each individual. Now you get randomly assigned the phenotype of a different individual. So every kind of association that is in there would now not be true, right? So because there is no relation now between x and y.
So, I'm sorry, what are you comparing?
The phenotype. Only the phenotype. And well, if I plot that again, I get a similar plot, just that there's not such a big difference anymore between the peaks and the non-peaks. Well, and now I again pull out the maximum, the largest squared beta, and I plot the same thing. Now there's again a regression line, which is, it's definitely not flat. But if we look at the individuals, we see that there's only one individual that has the mutation and the rest has 0. So that is something that probably just could happen at random, right? In this case, we know that it happened at random because there is definitely no relationship between the SNP and the phenotype. Is this clear, what I'm doing here? Okay, good. Okay, so for a comparison, this was what we observed before. So the regression function looked very similar, but here there's actually some individuals that, well, on average have a difference between the average of these. Here also there's a difference in average between these and these, just that we're taking the average over a single value here, which is a pretty boring average. And well, if you look at the histogram for these permuted value, now they're absolutely Gaussian. If we look at for comparison the histogram that we plotted before, it was not completely Gaussian. So there seems to be something that makes these random beta values look like a Gaussian. It's just an observation so far, right? if we plot them together, so now these are the permuted ones, that's the maximum likelihood fit, and that's the maximum likelihood fit to the real ones. We also see that, well, for those real ones, there are apparently more larger and smaller regression weights. So those distributions are clearly not the same. Well, this is just to plot the other way around. So if we take the distribution of the real ones and plot over it the distribution of the permuted ones, then they're very different. And if you look at, so for example, everything. So this area here would be the 5%. So this would be 2.5% of the mass of this here, and everything same here. And well, if we look at the distribution here, this would probably correspond to 5 to 10% of the mass under the observed values. So those distributions are very different, and they mostly differ in the tail and how much mass is in the tails. Well, so what does this theory say about this distribution? That's one question we can answer. And so the other thing that we also know is that here, if a value of beta is that large, then it's just completely random. It's just because it's just fluctuation in the maximum likelihood estimate. There is nothing that should systematically make the beta differ from zero, because you cannot systematically separate out like large individuals and small, or fast flowering and short flowering individuals by a SNP, because the flowering was assigned at random. So there cannot be any relationship. So this one gets small and large only by random. Where here there are probably a lot of mutations that differ between plants that flower early or that flower late. So while for many also there's probably no difference. I mean, if you just have a mutation that has nothing to do with flowering time, then you should get a value that looks like that. But if you have something that actually makes you flower earlier or later or not flower at all, then you should get a regression weight that is either very large or very small. So this is something. So this difference in the distributions is kind of what we hope to use to kind of set apart the mutations that matter from the ones that don't matter. Right? Is this conceptual idea clear?
Can you repeat the last part about just read some of the last part again?
Okay, so this is now, I mean, what's contained in this plot is it's a histogram over all the regression weights. So it's the regression weight for each. We take each measured SNP, and we see if we fit a regression function using that SNP from that SNP to the phenotype. Can we kind of, if we do this plotting individuals that don't have the mutation to individuals that have the mutation, can we separate out these two distributions? A beta of 0 basically means that we're plotting this flat line that the points that we measure on the right side are at the same space as the points that are on the left side. Right? But many of these, even if there's no information, even if this function, well, if we observed more data points, we probably would at some point, well, no, even if that function is not causal, well, it doesn't have to be causal, but if there's no systematic effect going that separates early from late flowering, we sometimes observe some of those regression functions that have a slope. But these we only observe it due to randomness because, well, just very few individuals, for example, have these mutations, and they happen to have a later flowering time than the average. And for that reason, we can draw such a line. And, well, for the true SNPs, most of those will also just have a random regression rate because there's no matter whatsoever between a mutation that lies somewhere in the genome where there's, for example, no gene or something like that, and the phenotype. But some of them also might lie, for example, in a gene that may, well, for example, makes you not flower at all, that makes you flower a little bit earlier, whatever. And for those, we'll systematically get larger regression functions. So this is probably kind of, this distribution here is kind of a mixture of this random distribution and something else. And so the interesting bit is what's different. And probably this something else is probably mostly located here, while the random part, well, should look like exactly like this, so it should mostly be located here in this part of the histogram.
So to choose our significance threshold where we decide this is causal, this is non-causal, we do permutation and see how much what we want as a false discovery rate. We use that as our threshold.
We could do that. I mean, so far we have only observed. Yeah, I mean, now we can use that, for example, as a recipe, as you said. Yeah, that's something that we could do. We could, for example, well, do this sort of permutation many times, such that this is really a very smooth function, and that well, we can either do a fit, or we can just use the values. We can just look at how many observations we have below a certain threshold. So, and then we can compare the value here to that distribution. If we are far out of the distribution, then probably we have something that is a true association, while if we observe something that lies here, then probably it's not a true association. So this kind of gives us a recipe to decide whether the regression function that we've just determined is a useful one or not. Whether we can report that result or not. So, well, what does the theory say? So let me quickly summarize. We saw a difference between these distributions. We saw that these looked like Gaussians. These kind of, but not quite, because it's kind of made the reasoning that is probably it has part of it is actually Gaussian distributed like this, part of it differently. Yeah. And the other thing that we observed is that just because the regression line has a slope doesn't mean that this is a useful regression function. So, well, let's look at the estimator again. So the estimator is, well, the pseudo-inverse times y. And we can write it differently. We can basically now write it as a sum over entries in y. So the regression rate is kind of a weighted sum. Well, this is not just one entry. It's the one entry that we're interested in for the SNP. It's now a weighted sum over the data samples. Data samples have some distribution that we don't know, right? But given these two things, can we say something about the distribution of the beta values that we expect? So that was the applied bit. And well, just to give you a short outlook of the question that we'll try to answer next week. View full screen mode. we look again at the Gaussian distribution. Well, let's do an experiment and throw dice n times. Let's measure the sum. Let's do that many times. What is the distribution of the sum? Well, if we throw the dice once, it's just like uniformly between 1 and 6. Twice, if some values between 2 and 12, if you throw it many times, we again observe the same thing as before, that something goes to a Gaussian distribution. So a different experiment. Let's just take samples from a uniform distribution between in the interval 0, 1. Do that many times and compute the mean. And well, the experiment is now get n samples, compute the mean of those n samples. That's one experiment. So we repeat that many times, and those are those distributions, right? So it's not just so basically here we threw the dice 5 times, but then and computed the sum, but did that many times, and this is what gave us this Gaussian-looking thing here. Now we do the same thing with the uniform distribution. Well, if you just get a single uniform number, take its mean, then it's obviously just the uniform number itself. If you do it with 2, it looks like that. If you do it with 10, then it already looks almost like a Gaussian. So this kind of gives us an idea that, well, if we, no matter kind of for very different distributions, we get something that if we sum many samples of those up, we will get something that looks like Gaussians. And this insight we'll use, for example, in order to look at the distribution of those regression weights, because those regression weights, as we said, are kind of a weighted sum or are a sum of all the samples, where we don't know the distribution of the samples in the first place, the true distribution. So we can also assume that under certain conditions we observe Gaussians, and that is what we observed on the permuted data. Okay, well, this is, well, quit for today. Yes, we go over it on Wednesday, but if you have questions, we can